Hey Dr. Sharma, this is Nitin Potturi and Carson Neal. We both hope you're doing well, and today we're going to give you our presentation on glioblastoma and its genetic factors.
So we're going to start this presentation today by talking about the basics of glioblastoma. So essentially, glioblastoma is a very aggressive type of cancer, and it forms in the brain on cells called astrocytes, which support the nerve cells in the brain. And this disease can affect anybody. It generally affects older individuals, but people of any age can suffer from it. The symptoms generally consist of headaches, nausea, vomiting, and seizures. And like I said, this is a type of brain cancer, with brain cancer being defined as the abnormal and uncontrolled growth of cells in a mass called a tumor in the brain. And this is a big problem because having any sort of mass in the brain can affect a lot of things because of how complicated and complex the brain is. And brain cancer is the 10th leading cause of cancer death in the United States, with glioblastoma being the most common form and also the most deadly. And although there's not a lot known about how brain cancer functions, they do know that the tumor suppressor gene TP53 has been found to be helpful in defending against malignant tumor growth in this brain cancer. And then glioblastoma is actually broken down into multiple grades of severity. And these levels of severity can require different treatments. Some of the lower grades can involve more regulation over time and just keeping an eye on it, whereas the higher grades require immediate intervention. So grade 1 is your pilocytic astrocytomas. These are less aggressive, relatively benign, often seen in pediatric patients that suffer from glioblastoma. And this puts pressure on the brain and causes symptoms because of that pressure. You have grade 2 gliomas, which is a slow-growing cancer. It can cause symptoms if it's not treated, and also if it's not treated, it can progress into a grade 3 or 4. With a grade 3 glioma being malignant, it's composed of anaplastic cells, which means that they no longer function like the cells that they derive from. It's kind of like the non-differentiated cancerous cells. These tumors are pretty aggressive. They need to be treated immediately, or else they could turn into a grade 4 and they could be deadly. Grade 4 gliomas are called glioblastoma multiforme. They are very aggressive. They can spread easily. Sadly, this is the most common form, also the most dangerous, and without quick treatment, it can quickly lead to death. The current treatments for glioblastoma are not very particular to the actual disease. They're more general solutions to fix all different types of cancer. As you can see here, glioblastoma does not really have a very good survival rate. The National Cancer Institute estimates that 15,500 of the 23,000 people diagnosed with brain or nervous system cancer died, and that was in 2015. And like I said, they don't really have treatments that are very specific to fixing this disease, and most of the time they end up going the surgical route. They'll map out where the tumors are using MRI and CT scans, and they'll try to go in and remove that, although because of the complex nature of the brain, it's very difficult to remove the entire tumor and the surrounding malignant tissues, so oftentimes they have to go through radiation or chemotherapy afterwards to try to eradicate anything that was left behind. There are more treatments that are being looked into, but the treatments that have been discovered in the past 3 decades have only added a median of about 3 months to the life of glioblastoma patients, so they're really looking for better treatments.
Both of us were really interested in our investigation on glioblastoma, so we wanted to dive deep into the genes of glioblastoma and talk about those in depth. So we decided to use online databases in our investigation. For example, we used the National Center for Biotechnology, NCBI, which is a publicly available dataset source. And what we did is just searched up publicly available datasets through this website, and we used those and analyzed those to get our data and analyze that information later on. And Gene Expression Omnibus is a subdivision of NCBI, and we used that alongside GO2R, which is just like a part of Gene Expression Omnibus. we use both of those to thoroughly analyze the data sets that we acquired through NCBI. And then StringDB is separate from both of these, but it's also publicly available. And what this does is it helps you characterize individual genes based on their families and functional partners, and that's what helped us further understand what each gene that we selected was doing in each scenario. So through NCBI, we used the dataset GSE 31095, and this specific dataset had 20 total patients, 12 healthy and 8 patients with glioma. And on the right, you can see this dataset that we got from analyzing the dataset through Geo, and it shows on the right, the red is are the 8 glioma patients, and the blue are the 12 healthy patients, which are the control. And you can see that all the reds are lower than each of the blues. So the analysis of the top 250 genes is something that we also did through Geo. So we can analyze the data set through Geo and get a massive list of all the genes that are either upregulated or downregulated for the glioma patients in relation to the control patients. And from that, we were able to make a string database, which is shown on the right. And we selected 4 individual genes from this data set through string to analyze further. First of all, we chose ISG15, then we chose HSPI1, RPL5, and RPL23. So the chart you see on the bottom, the important thing to note here is the log FC value on the right. And the log FC value is the log fold change. So what this means is the value you see here, it's negative, which means that the glioma patients have lower values or lower regulation of specific genes in relation to the healthy patients. So if there's a log FC value of 0, that means that there's no difference between the control and the affected groups. But in this case, since it's non-zero, then that means that there is a change. So the first gene we're going to talk about is ISG15, and we can see on the chart on the bottom right that we also got through Geo that this specific gene is downregulated in glioma patients in correspondence to the control groups. So what this gene does is it increases effect of cisplatin on cancerous tumor cells, and cisplatin is currently a common drug used to treat cancer patients to attack the tumor, we could say. And since this specific gene is downregulated, it makes cisplatin less effective, which means that tumor cells can grow a lot easier than they could on control patients without glioma. So through examination of the specific gene, we were able to understand that this gene is important to highlight when trying to develop a cure for glioblastoma. And you can see in the diagram on the top right of the slide, you can see the string database diagram of this specific gene. So what this diagram means is that it connects ISG15, which is the red bubble you can see right there. It connects the gene with different genes in its family which produce similar proteins, and you can see all the connections between it. And what it means is that since just this gene is downregulated and it isn't functioning as well as it should be in relation to the control patients, the whole family itself is weakened because of that one gene not working. The next gene we're going to talk about is HSPE1, and this specific gene codes for HSP60, which is a heat shock protein, and this protein binds to other proteins to ensure that they're folded correctly. And if they're not folded correctly, they're going to break that protein down. And you can see again in the chart on the bottom right that the glioma patients on the red side have severely downregulated protein production from the HSPI1 gene. So we think because of the downregulation, it's probable that the incorrectly folded proteins are going to be used in the cell, even though they shouldn't be being used, and that could somehow impact the cell proliferation, but we can't say this for sure since research hasn't been done on this yet. But we think that if further research is done, something will show up that can point to something similar. And again, we can see in the top right that this gene not working correctly impacts a ton of other genes, which all in all will just shut a lot of other things down.
There are two other genes that are also downregulated based on this data set. In the bottom right corner, you can see the graphs that show healthy patients versus glioblastoma patients and their levels of the proteins that come from these genes. In the top right corner, you can see the gene family that these come from, which helps show the other genes that can be affected by the downregulation of these genes. And so the function of these genes and their proteins are to aid and assist in ribosomal assembly and protein translation. But there's research that shows that some of the proteins will activate the p53 tumor suppression pathway when subjected to ribosomal stress. But the problem here is if these are downregulated, then the ribosomal stress is suppressed, which means that the RPL5 and RPL23 will not be released and they will not be able to perform their duties when it comes to p53. If these were released, they'd be able to inhibit p53 ubiquitination and degradation, and basically without them the p53 degrades and it is targeted in that way, which leads to less regulation of tumor growth. So basically if this wasn't downregulated, then those proteins would be able to come out, they'd be able to inhibit the degradation of p53, and it would lead to cell cycle and proliferation arrest, which would completely halt the cancerous growth. So it's very important that these are downregulated and that be changed. As far as treatment, so I discussed what the current treatment is earlier. Now I'm going to talk about kind of what we think the treatment should be geared toward based on this data. And so as you can see here, these major genes that we talked about are all downregulated. So in order to raise the amount of each of these genes back to normal, we would suggest a method which the rate of gene production can increase and therefore upregulate the specific genes to normal levels. And being college students, we don't exactly have the correct answer for that, although I do think that that's the direction it should be going. One suggestion we have is that we could use enhancers or cis regulatory modules, which could lower the ratio at which the RNA polymerase II can allow the process of gene transcription, which would increase the overall genetic expression for those specific genes and hopefully allow the patient's bodies to fight off glioblastoma more effectively.
So through our investigation, we found out that these four genes, HSPE1, RPL5 and 23, and ISG15 are all primary causes or significant causes, factors, for the growth of brain cancer gliomas in affected patients. And we can tell this because all four of these genes are downregulated and all four of them are correlated to something that could definitely lead to the growth of glioma cells. And we think that with the correct enhancers, we could strengthen the genetic makeup of patients with glioma in the future. And with enhancement treatment, enhancement of transcription will allow for patient-specific treatment that will help to increase expression of crucial genes, which will reduce the effects of glioma. So we think that if we use enhancers, we can upregulate the previously downregulated genes and at least put them at normal levels so that the patients that may get glioma will have less of a chance of getting glioma.